Well, I think we'll start. A couple things I want to do today. One is to review the schedule with you again. This week, we have a lecture today on molecular genetics, and on Wednesday, we'll talk about climate change. And then Monday, a week from today, is test 4. Test 4 will cover everything that has not been covered yet. In other words, everything that we've covered since test 3. And then next Wednesday, the Wednesday after that, is when you'll present your cystic fibrosis projects. Then the next thing after that is the final, which is a week later, June the 2nd.
Woo, final, woo.
That's it. Okay, the next thing I want to do is go over quiz 8. Share my screen with you. Do you see quiz 8?
Yeah, we see it.
I'll make it bigger. Just want to go over the answers. The first question, anaerobic respiration leads to ethanol production, commonly occurs in what biological entities? And the answer is yeast, or if you said microorganisms or even bacteria, you got credit for that because all of those are true. Alternatively, if anaerobic respiration leads to lactic acid production, it could result in sore muscles or anything equivalent to sore muscles, a burning sensation, anything that we had talked about in class. And the basis for these questions are from these illustrations from the lecture, from the ethanol production under anaerobic conditions, and the sore muscles from lactate accumulation in muscles. Okay, the second question, choose the correct sequence of steps in glycolysis and oxidative phosphorylation. Well, that question derives directly from this illustration. And the correct answer is that based on the question that's asked, you start with glucose, and the next step is pyruvate, which goes into the mitochondrion, and that's converted to acetyl-CoA, which is the first step in the Krebs cycle. So Krebs cycle would be next. Then protons are produced from the Krebs cycle, and electrons, which go into the electron transport chain, and that leads to chemiosmosis, which makes ATP. So that's the correct sequence of events directly from this particular picture. Third question, a chloroplast light reaction starts with the splitting of water into molecular oxygen and protons. And that came from this illustration where the first step here is water being split into molecular oxygen and protons. The electrons that are generated flow through first photosystem 2 here, and then to photosystem 1, and end up in the chloroplast dark reaction, providing energy for the Kelvin cycle to fix gaseous carbon dioxide into solid sugar. And that is based on this, where I emphasized carbon fixation, and that this enzyme Rubisco is responsible for taking a gas and converting it into a solid sugar. And then number 4, under anaerobic conditions, what happens to the Krebs cycle? The answer is it would stop. And if you go back to this illustration, the end of electron transport is the final electron acceptor is oxygen. If there is no oxygen, no electrons are accepted, and this entire process stops. Electron transport chain stops, and the process backs up so that the whole system stops. And it converts to pyruvate being converted into either ethanol or lactic acid. So from here on, it's simply gone under anaerobic conditions. Number 5, acetyl-CoA is a critical part of oxidative phosphorylation because it is the first step in the Krebs cycle. If you were starving, acetyl-CoA would be generated from. The answer is oxidation of fatty acids. And again, if we go back to this illustration, so if you're starving, you're getting no input of sugar. So where would the energy come from? From the oxidation of fat to fatty acids to acetyl-CoA. So that's if you were starving. And the next part of the question is, what if you were gorging on pasta, so you're getting a lot of carbohydrate, then the other side of the pathway would run from glucose to acetyl-CoA. So the two answers is a two-part question. The first is oxidation of fatty acids, and the second is pyruvate. From among the possible choices here, pyruvate was the most appropriate one. And finally, match each term with the best fit description. A photosystem 1 is absorption of a photon, and we've looked at that illustration already. Gas to solid conversion, that's CO2 to sugar, is the enzyme Rubisco. Grana are stacks of thylakoids. If you remember this illustration, these are grana. They're stacks of thylakoids. Stroma is the dark reaction. Again, this stroma here, the dark reaction, the Kelvin cycle. Stroma being the spaces between the thylakoids, between the grana. Mesophyll cell, abundant chloroplasts. You see that here. Anatomy of a leaf. The mesophyll cells are the ones that have the majority of chloroplasts. And finally, a proton gradient with chemiosmosis. And there are a lot of illustrations of that. This is one example, chemiosmosis. And a proton gradient. What's important here is that the concentration of protons on one side of the membrane is much higher than on the other side, so that passive diffusion allows ATP to be synthesized. So any questions about any of these answers, or about the questions, or why they were asked? I'll put this set in Canvas so you can refer to it as you wish as a study guide. Can you see the screen, molecular genetics?
Yes.
Okay. Well, we spent a lot of time on Mendelian genetics. But so far have only referred tangentially to molecular genetics. There were a number of issues that have come up, which some knowledge of molecular genetics would be useful. So what I've done for today is chosen certain topics that have been revolutionary, have changed biology and consequently had impacts worldwide. And the topics that I've chosen that you should know about because they affect you and have affected you, and you read about them in the news media frequently. And these are restriction endonucleases, polymerase chain reaction, PCR, we've talked about that before, gene therapy, just very briefly, and then genetically modified organisms. And I'll briefly review CRISPR because CRISPR is behind a lot of what we have going forward. Okay, starting with restriction endonucleases. The 1978 Nobel Prize was awarded to these three guys because they discovered endonucleases, which was quite a feat. And they did that at a time when, first of all, nobody had any idea that restriction endonucleases existed. And because of that, they had trouble getting money to fund their research because people didn't think, everybody knew about nucleases. Nobody thought that going further than that would be of much interest. So I want to put this restriction endonucleases in context, starting with the food that we eat. Food is largely composed of carbohydrates, fats, proteins, and nucleic acids. And these are all depolymerized enzymatically. So carbohydrates, there are carbohydrases. For fats, there are lipases. Proteins have proteases, and nucleic acids have nucleases. Now, among all these polymerases, only the nucleases can have site specificity. The others do not. And let me explain on this slide what I mean by that. So first consider proteases, which act on proteins. So here you have the yellow balls, an example of the amino acid chain in a protein. And there are different types of protease. Carboxypeptidases start at the carboxyl end and nibble off amino acids. Aminopeptidases start at the amino end and nibble off amino acids. Endopeptidases cut in the middle and make shorter and shorter peptides, which then can be converted to amino acids by the other two. And so this is pretty much a random occurrence. There's no specific sites in an amino acid chain where a protease will cut. The lipases start with fat and it's converted to free fatty acids. Again, there's no specificity in site selection. And similarly, the carbohydrases, and the example here is cellulose from plants, these long chains of glucose. And there are different types of enzymes that break up cellulose. So there are the exoglucanases, which nibble off sugars from the non-reducing end. There are exoglucanases, that start at the other end, the reducing end, and take off sugars 2 at a time. And there are endoglucanases that cut in the interior of the molecule. And finally, there are beta glucosidases, which cut the maltose molecules into monomers of glucose. So again, there's no site specificity here. These are random choices. I want to contrast the randomness of these polymerases with the nucleases. They can be either specific or non-specific. So starting here on the left, non-specific nucleases are either endonucleases. And if you look at the bottom left here, an endonuclease cuts the amino acid somewhere in the middle. Exonucleases cleave nucleotides from either the 3' or the 5' end. So here you see an exonuclease. And then there are specific restriction endonucleases, which cut the lower left here. They cut at very specific sites, specific nucleotide sequences. And of these, first let me say something about these restriction endonucleases. They're found only in bacteria. Eukaryotes don't have them. The reason bacteria do is that it's sort of a defense mechanism against viruses. So when a virus nucleic acid enters a bacterial cell, these endonucleases will chop it up, cutting at their specific sites. And the sites are specific so that the bacteria don't chop up their own DNA. And in fact, in case the bacteria do have sites for their own enzymes, those sites are methylated so that they're protected. They're not cut by the restriction enzymes. Well, the cuts that these endonucleases make are of two types. They're shown here in the center, the color. A sticky end enzyme cuts with a staggered, in a staggered fashion so that there is an overhang left on both strands. Whereas a blunt end enzyme cuts without an overhang so that the edges are uniform. There are no overhangs. And so it gets even more detailed than that. We have the non-specific and the specific. And among the specific, there are several types. Type 1, these are enzymes that have target sites in the DNA, but they don't cut at their recognition site. They cut about a kilobase away in sort of a non-specific fashion. The type 2 restriction enzymes I have here in red because they are the ones that are useful in genetic engineering. Their recognition sites are palindromes of 4 to 8 nucleotides. And critically, they cut at the same site that they recognize. And they can have either sticky ends or blunt ends, and approximately 3,500 type 2 restriction enzymes are known. Type 3 recognize 2 separate inverse repeats, and they cut 20 to 30 base pairs away from their recognition site, so that's not very useful. Type 4 enzymes recognize methylated DNA, so if DNA is protected by methylation, they can ignore that and make a cut anyway. Type 5 enzymes I've highlighted because they use guide RNAs to target specific non-palindromic sequences found on invading organisms. And then they cut DNAs of variable length. And the reason I've highlighted this is that an example of a type 5 restriction enzyme is Cas9, which is an essential component of the CRISPR procedure. And I'll review that very briefly a little later. And finally, although it's not a restriction endonuclease, DNA ligase is always associated with endonucleases because it's a DNA joining enzyme. You remember from when we talked about DNA replication, the very last step was DNA ligase, which sealed the gaps. We have the same thing that is necessary in the cloning of DNA. The last step is always DNA ligase. Okay, so let me tell you a little more that you should know about restriction endonucleases. The length of the site, the recognition site, can vary from 4 to 8 nucleotides. And so the shorter the recognition site, the more frequently there will be recognition sites in the DNA sequence. So for a 4 cutter, every 256 nucleotides, there's another site. Whereas if you go to an 8 cutter, it's 65,000 base pairs between sites. And there are examples here of enzymes that do that. There's a list here of various different restriction enzymes and their recognition sequence, the source, the bacteria that produce them, and the type of end that they leave after cutting. It's important that you know that all restriction recognition sites are palindromes. What is a palindrome? It's a sequence of anything that reads the same forward or backward. So if you look at the recognition sequence of EcoR1 from E. coli, on the one strand it reads GAATTC, and on the other strand, if you read it backward, it's GAATCC, TTTC. And you can go through all of these, they're all the same. They're all palindromes. And one way that you can remember that, here are examples of palindromes, except with letters instead of the symbols. So a man, a plan, a canal, Panama. Read that backwards. Do you see the effect? You start with A on the right, A man, A plan, A canal, Panama.
The same. Same.
Another example, Abel was I, ere I saw Elba. Again, if you read it backwards, it says exactly the same thing. So that's a palindrome. The other thing we have to talk about is plasmids. What is a plasmid? Let me just go forward. What is a plasmid? It's a little circle, usually a circle, not always, of DNA. So here are the characteristics. It's a DNA molecule. It's always, not always, but usually found in bacteria. It replicates independently of the bacterial chromosome. Remember, the bacterial DNA is in a nucleoid, And plasmids are outside of that and replicate independently of it. They're usually circular and they're essential for DNA cloning. And here's an example of a plasmid for cloning. We have an antibiotic resistance gene, a selectable marker, usually what's called a polycloning site, a whole bunch of restriction enzyme sites here that can be cloned into. So that's a plasmid. Their functions are for toxin production or antibiotic resistance, but they're useful for cloning. And this illustration on the right shows you how that's done. So if you take a plasmid, shown here the red circle, that has a cleavage site, which in this case, let's say it makes a staggered end, cleavage by EcoR1. Okay, the overhangs, if you cut with EcoR1, are TAA on the one strand and AATT on the other strand. If you look over here at EcoR1, you can see that on one strand, the overhang is AATT. On the other hand, it's TTAA. So these are the so-called sticky ends. Now you've made that cut and you want to clone something into that cut. So you have foreign DNA. This could be DNA from a genome of some organism. So you cut this foreign DNA with the same enzyme, EcoR1. So you get the same sticky ends, AATT and TTAA. Each fragment has the same sticky ends so that they anneal, they bind. Remember, A's always bind to T's, T's always bind to A's. They bind to their complements so that you now have the plasmid with its gap filled, except for the ends. And that's where DNA ligase is used to fill in those gaps. So now you have a complete chimeric plasmid. So that's why plasmids are so important for molecular biology in general. Now I want to say a word about the naming of restriction enzymes. They have these exotic sounding names. Where do those come from? So the name consists of two components, 3 letters that correspond to the scientific name of the microorganism that produced the enzyme. So for example, if it's from Escherichia coli, its name is ECO, ECO. If it's from Haemophilus influenzae, its name is HIN, H-I-N, H-I-N. So that you can see that every restriction enzyme has a unique name based on the scientific name of the microorganism, plus a symbol that identifies which particular enzyme it is. So each name then is unique. EcoR1, there's also an EcoR2, an EcoR5, and I think several others from E. coli. From Bacillus, there's BAMH1, and then a number of other enzymes from Bacillus, and so on. So is that clear how these restriction endonucleases get their names? Because if you don't understand that, it can be pretty confusing.
Yeah, it's clear, teacher.
Okay.
I have a quick question.
Yeah.
So do we have to, are we able to synthesize these enzymes, or do we have to harvest them from these bacteria?
You buy them from lab supply companies now. In the early days, when they were first discovered, we prepared them in the lab ourselves. So we would take, for example, E. coli and grind it up and extract EcoR1 from it. And it was such a laborious process that laboratories would specialize. One would say, okay, I'll do EcoR1, and the next one would say, okay, I'll do BAM. And then we'd trade. But that's not necessary anymore. Now you just open your catalog and order as much as you want.
Okay, so then the big companies are synthesizing it, and they're not grinding up bacteria to get it.
No, I think they're grinding up bacteria. I think these enzymes are too big and too complicated to actually synthesize de novo. I don't know that for sure, but let me just go back. Here's a three-dimensional structure of EcoR1 showing where it cuts, its cut sites, so the purple balls. I think it would be pretty difficult to make that, although, as I said, I don't know for sure.
Right. Okay. I was just curious.
Just a quick question.
Uh-huh.
The last when Escherichia coli RI. So Escherichia coli is the name of the bacteria. RI is the name of the enzyme.
Yes. Yeah. So let's go back.
Yeah.
Yeah. Eco comes from Escherichia coli, eco, and then Escherichia coli makes several different restriction endonucleases. So R1 indicates one of those.
Yes.
And then,
Yes.
so
Escherichia coli is the name of the bacteria. We take that before.
yeah, that's the name of the bacterium.
Yeah, and RI is the name of enzyme, and HI is the name of the enzyme that's make difference.
Right, right. That's the symbol following the name indicates which enzyme it is.
Oh yeah, thank you.
All right, then one last thing. Based on everything I've told you so far, how is cloning actually done? And I've got here an example of how you would make a library of DNA from the genome of an organism. So it could be any organism, it could be bacteria, it could be mice, it could be a plant, it could be human, but you take, you want a representation of all the genes in that genome that code for proteins. So the steps in the procedure are listed here on the left. So remember that the genome of most eukaryotes is mostly non-coding, not non-coding for protein. So 95% or more of the genome is stuff you don't want. So how do you avoid cloning that? Well, you do it by, instead of isolating DNA from your organism, you isolate messenger RNA, because these messages are accurate representation of genes that code for proteins. So all the rest of the DNA that doesn't code for proteins is not represented by mRNA. Then you use this enzyme reverse transcriptase, which we've talked about before, which instead of going from DNA to RNA, it goes from RNA to DNA. And so you make a complementary strand of DNA. And it has to be DNA because that's what your vector is. The vector is made of DNA, and you can't clone RNA into DNA. So then this complementary DNA is digested with a specific enzyme, for example, EcoR1. And then the vector that you want to clone into is also digested with the same enzyme so that you have sticky ends. The ends of the DNA you want to clone stick to the ends of the vectors. And you ligate them together. And now you have these chimeric plasmids, and each one is different. Each one has a different piece. So all the vector molecules are the same, but the insert on each one is different, a different piece of the genomic DNA. These are readily transformed into cells of a bacterium, usually E. coli, where it's multiplied. E. coli makes a lot of plasmid DNA. You isolate that, and then from each cell, you have a clone, and that you can grow as much of it as you want. So if you had 1,000 test tubes, each with a unique clone in it, that would represent your genomic, the genome that you've just cloned into a vector. Then you can sort through those clones looking for any particular sequence you might be interested in. So that's how you construct a genomic library. Now, what are some of the uses of restriction enzymes? Well, for recombinant DNA, I've just showed you how that you can clone any piece of DNA can be combined with any other piece of DNA. Using that technology, you can make genomic libraries, you can make restriction maps, and DNA fingerprints, and then the list goes on and on and on. So let me just illustrate for you how these things are done. So if you want to look, first let me start here with a restriction map of a virus, the phage lambda. So these lines here represent where enzymes cut. So if you look at the first one, it's BAMH1. Where does BAMH1 cut? 1, 2, 3, 4, has 5 sites in the lambda genome. The next one down is EcoR1. It has 1, 2, 3, 4, 5 sites. But you can see they're all different. BAM and ECO cut the same number of times, but at different places. So if you then want to know, if you then want to visualize or actually isolate these various fragments, you do that with a process called gel electrophoresis using a polyacrylamide gel. And this is the way it's done. You melt some polyacrylamide, pour it into a mold. You take your samples, load them into little holes in the now solidified polyacrylamide, and then you hook it up to electricity. Now, DNA is negatively charged. So it will migrate, if you have an apparatus that has a negative end and a positive end, DNA will migrate toward the positive end. And the rate at which it migrates is a function of its size. So the bigger the molecule, the slower it goes. And the little molecules go really fast. So you'll end up with an array of bands in a gel. And that's shown here in the middle called Lambda Digest. So this is a picture of an actual gel of lambda DNA that's cut with either EcoR1 or BAMH1 or both. So if you look at the bands and count them, A and F are together. F is really down here, but here's 1, 2, 3, 4, 5, 6. And in an EcoR1 digest, there are 1, 2, 3, 4, 5, 6 fragments. And the same is true for BAM. There are 6 fragments, 1, F is the same, so it's 1, 2, 3, 4, 5, 6. And if you actually were interested in cloning one of these bands, you can cut it out of the gel, elute it, and clone it in a plasmid. So is it clear how you do, first of all, a restriction map? It's simply a map of where the restriction sites are on a piece of DNA. And then if you cut at those sites, you can display the fragments and actually isolate the fragments using gel electrophoresis. Now, and that's useful in many, many ways. And an example I'm going to show you here is in forensics. So, and we're going to look at a video in a minute about this, but here there's a rape investigation. And here's DNA fragments from the victim. There are 3 suspects, A, B, and C. And this is spelled wrong. This is evidence from the site. So they got DNA from the site of the crime, and it matches up precisely with suspect A. So that unequivocally identifies suspect A as the one who did the crime. Now, I want to stop sharing that and show you a video.
Thanks for stopping by. This is 2 Minute Classroom, and today we're talking about polymerase chain reaction, or PCR as it's more commonly known. First, let's talk about what PCR is and how it works, and then I'll give some examples of what PCR is used for. PCR is a technique used to amplify or copy small specific segments of DNA. For PCR to be successful, you need the DNA sample you want copied, DNA primers to identify the specific sequence of DNA you want to amplify, lots of raw nucleotides to form the DNA copies, and a heat-resistant DNA polymerase to make the new copies. This all goes into a special, stable solution. Once all the ingredients are in, you run the solution through many temperature cycles until you reach the amount of DNA copies needed. There are 3 steps to this cycle. Step 1 is denaturation. The solution is heated, allowing complementary DNA strands to separate so that all the DNA in solution is single-stranded. Step 2 is annealing. The solution is cooled so that the DNA primers can bind to the specific region of DNA you are interested in. Step 3 is extension. The temperature is raised so that the DNA polymerase can bind and perform the necessary operation of synthesizing new DNA strands. This cycle is repeated about 30 times, takes about 3 hours, and creates over 1 billion copies. So what is PCR actually used for? PCR has many research-based and practical applications. It's used for DNA mapping, like in the Human Genome Project. It's used to check sections of DNA for genetic testing to determine potential disorders. Companies like 23andMe and Ancestry.com use PCR to run personal DNA tests. And my favorite application for PCR is in forensics. Whenever a crime is committed, the perpetrator almost always leaves their DNA at the crime scene. This could be hair, semen, skin, or even blood, for example. Investigators search for this evidence and, thanks to PCR, can amplify the DNA and compare it to the DNA of suspects. This genetic information is then put into the FBI's DNA database, CODIS. But it gets even better. Investigators have been able to take DNA evidence from crimes committed long before CODIS was put together and run it through the current system. This has solved many cold cases and also exonerated prisoners who have been wrongfully accused, even some that were on death row. Needless to say, PCR is one of the most important scientific advances ever. And now you know why. If you have additional questions, I'll do my best.
Okay, can you see the screen again?
Yes.
Okay. That video was an introduction to PCR. So we've finished up our restriction endonucleases and talked about forensics. Now I'd like to introduce you to PCR because it's ubiquitous. It's found everywhere. What is it? It's the synthesis of large quantities of a specific DNA segment, starting with as little as 1 molecule. So that's really the big merit of PCR, is that you can start, you have to have at least 1 molecule, but 1 is enough. So that's why you can go, for example, into a crime scene, and if you can find even 1 molecule of DNA from the criminal, that's enough to produce as much of that DNA as you want. So how is it done? The video went through the components of PCR. I have them here again. You have to have a target DNA template, something that you're trying to amplify. Then it's critically important that you have DNA polymerase, a particular DNA polymerase called TAC1, Taq polymerase. It's from a bacterium from a hot spring, so it's very heat tolerant, does not denature even at boiling temperatures, and its optimum for catalysis is at about 70 or 75 degrees. So you need that enzyme, that's critical. And if you look over here on the right, this is the reaction mixture that you need to do PCR. So you need the template, that's here. Here's your genomic DNA, there's one little piece of it you want to amplify. That's the target. And now you put that into a mixture of this enzyme Taq polymerase plus primers. Here are the primers for the ends of your target. Then you need the triphosphates because you're going to be synthesizing DNA. So you have to have the A's and T's and G's and C's that are needed to make DNA. And then you need a buffer system. So the procedure, it was described in the video. First, you heat to a very high temperature, usually about 95 degrees. So that's shown here in step 1. And what does the heat do? It breaks the hydrogen bonds so that the 2 strands of DNA in your target can separate. Then, after you've separated them for a few seconds, you cool to a lower temperature, 50 or 60 degrees, so that the primers can anneal to the ends of your target. Then step 3 is to raise the temperature to the optimum for Taq polymerase to about 75 degrees, so that Taq polymerase can synthesize DNA corresponding to the target. So now what you've done, if you started with 1 molecule of target sequence, you've gone through 3 steps, which took a few minutes, and now you have two molecules of the target. But you don't stop there. You do it again. Again, you heat, you cool, extend, and now you have 4 molecules. Started with 1, now you have 4. You keep doing that. Cycle 3, after you do cycle 3, now you have 8 molecules of your target. And if you keep going, cycle 5, you have 32 copies. Cycle 15, you have 32,000 copies. And after cycle 25, you have 33 billion copies. And so in a matter of an hour or so, you can go from 1 molecule to a whole test tube full of DNA, specifically of your target. And this is the beauty of PCR. That's why it is so widely used. When we talk about screening for coronavirus, most of the screening tests are based on PCR. Now, we've looked at the video already. So I want to tell you a little bit about the history of PCR. It's a very strange set of circumstances that led to the discovery of it. And there's a question, if it hadn't been for this one guy, Kary Mullis, would PCR ever have been invented? So going back prior to PCR, the 1980s and before, the only way to get DNA was to isolate it from an organism that made it. And this in a lab was usually an E. coli. But then in 1969, a man named Thomas Bruck isolated this new species of bacterium from a hot spring. And he called it, the bacterium was Thermus aquaticus. And it produces this enzyme, Taq polymerase, which can stand near boiling temperatures without denaturing. And its optimum for catalysis is about 75. So this is a very unusual enzyme, very unusual DNA polymerase. And it's essential, it's critical for doing PCR. So it was only by chance that this guy isolated this bacterium and discovered the enzyme. Now you can buy it from lab supply companies. Then in 1979, there was this new biotech company in San Francisco. They hired a guy named Kary Mullis to do a specific job. In 1986, Mullis presented a paper at a symposium in Cold Spring Harbor. And this was the first time that PCR was presented in public. Then that same year, Mullis left CETUS. And in 1993, he got the Nobel Prize for this because people immediately recognized, immediately started to use PCR in their work. And an interesting side note here, when you get the Nobel Prize, you have to go to Stockholm and make a speech. And in Kary Mullis's speech, he described described how he proved that PCR actually worked. And that happened one night. It was the same night that his girlfriend left him because he didn't pay enough attention to her, spent all his time working on PCR. Financially, when CETUS, when Mullis left CETUS, they gave him a $10,000 bonus for his Nobel Prize winning work. Then they turned around and sold the technology to Roche, a pharmaceutical company, for $330 million. So Kary Mullis really didn't benefit much financially from doing this. So applications of PCR, I think it's pretty obvious, all the different types of matters that PCR can deal with. But in general, it's used whenever a large amount of DNA is needed and the amount of target DNA is small, as little as 1 molecule. So for example, I've already mentioned it's used as one of the testing procedures for coronavirus. It is, I think, the standard test of the CDC. Can use to identify mutations in disease. We've talked about forensic. Any source of DNA from blood, hair, semen, etc., can identify a criminal unequivocally and prove either innocence or guilt. We talked about Neanderthal DNA. and about this man, Savante Pabo, and how, when I asked the question, how did Pabo do it? And then I had PCR, it's because Pabo is entirely dependent on PCR to amplify DNA from Neanderthal bones. So as I said, if he could get even one molecule of DNA from a bone, he could amplify it. He could make as much of it as he wanted. And there are many other ways. It's used daily in all molecular genetics labs. So the critical points in PCR development were the discovery of Taq polymerase. This is an enzyme that's absolutely essential for PCR, and the hiring of Kary Mullis by CETUS. And then they didn't pay any attention to him. Instead of doing his job, he worked on the polymerase chain reaction. So I don't have time. I don't want to go through all the weird things. Well, maybe I will. Kary Mullis was a very, very strange guy. He had a PhD in biochemistry, he got from UC Berkeley, but he also wrote fiction. He was a bakery manager. And after he invented PCR, he went on to do other things, such as he used PCR to amplify DNA from deceased people, like Elvis Presley, Marilyn Monroe. And then he embedded the DNA that he made from these people and sold it as jewelry. He was a conspiracy theorist. So for example, he didn't believe in climate change. He didn't think there was an ozone hole in the atmosphere. He didn't think that HIV causes AIDS, among other things. He was a kind of iconoclast. He didn't believe in anything conventional. And he was a champion of hallucinogenic drugs. He claimed that LSD gave him an insight into how to do PCR. He died in 2019 at the age of 74. And he said a lot of crazy stuff, like this last one here: science as it is practiced today is probably very similar to what was called science in 1634. I don't even know what that means, but it's something that he said in front of an audience. So my point here is that this really important application was developed by a guy that was a very strange character, and just illustrates scientific advances can come from anywhere. Interestingly, that PCR was his only scientific contribution. He didn't do anything of consequence before that or after that, at least not in science. I'm not going to say much about gene therapy. It's the delivery of nucleic acid into a patient's cells as though it were a drug. That's to treat disease. The idea is you treat the cause of disease rather than the symptoms. And so approaches to doing this: you can replace mutated genes, or you can fix them, or you can make disease cells more evident to the immune system. And I have here illustration of how gene addition and gene repair work. So if you have a vector and have cloned in that vector a functional gene, you can take a cell that has a mutation and integrate that gene so that now you have a heterozygote. You have a mutated copy and you have a wild type copy. And you hope, excuse me, you hope that the wild type copy is the one that wins out. Gene repair is a variation of that, or again, you have a DNA template, the wild type, in a viral vector. You put that into a cell that has the mutated chromosome, and then you have a recombination event, a double crossover, so that you replace the mutation with the wild type sequence. And now you don't have to worry about the mutation at all. You have the normal gene product. And if you think about this in terms of your project with cystic fibrosis, that's a genetic disease. So if you have a mutation in the CFTR gene, if you were to replace that with the wild type, now you don't need a drug. You've got the wild type gene and the mutation is gone. And I want to remind you of CRISPR. Now, you've seen all these slides before, so I'm not going to go through them in detail. I just want to remind you that CRISPR is a relatively new technology, and it is in and of itself revolutionary, the genome editing technology. So this is if you want to make a specific mutation. Why is it so important? It's because CRISPR permits any mutation to be made in any organism. It's site-specific, as you can deal specifically with any site in the genome, and it's universal in that it works in any organism, from bacteria to plants to animals. I don't know of any organism that's ever been tried that it didn't work. Just to remind you how CRISPR works, it's very simple conceptually. It's based on a guide RNA and an enzyme called Cas9. What is the guide RNA? It's an RNA molecule that's synthesized in the lab to correspond to a target. So there's a gene in your organism that you want to mutate. You make a guide RNA that corresponds to that gene, and then you mix that guide RNA with the enzyme Cas9. So the guide RNA takes Cas9 to the target site. And you get a matchup, complementary sequences. And when that occurs, Cas9 cuts the DNA at the end of the target DNA. And now that this site-specific cut has been made, you can do anything with it you want. You can put another piece of DNA there. You can switch pieces around. There are any number of different types of mutations that you can make at this site-specific cut. So conceptually, it's very simple. I'm told that even high school students now do CRISPR in their high school labs.
Excuse me, Professor, I have a question related to the previous slide. In these parts, the cuts of DNA that replaced from everywhere, how should they find it from the from other organism, or they are synthetic?
You mean the source of the target?
Yeah.
The source of the target can be anything. It could be DNA from you. It could be from a plant or an animal or a bacterium. The only requirement is that it's DNA.
Okay.
So DNA can come from any source and be a target for CRISPR.
Okay. But the exact result of this trial, what's the result of this trial? making a mutation for what?
Well, supposing you wanted to eliminate the mutation in a CFTR gene, you could use CRISPR to do that. You could just cut out the piece of DNA where the mutation is and replace it with wild type sequence.
Okay, okay, thank you.
So does the Cas9 cut it out? Because the way I was understanding it is it just creates that staggered cut in sort of one place, so to speak. So would you need two markers then? So that way then it would actually cut out that entire section?
It just makes a cut. That's all it does, is make a cut. So if you want to delete something, you have to use another enzyme to make another cut to delete what you don't want. So all
Gotcha.
Cas9 does is make a double strand cut.
Gotcha. Okay. That was what I thought I was doing. I was just a little. Okay.
Yeah,
So
I actually, go ahead.
I was just going to say, so theoretically you could deactivate a gene by cutting it in the middle and then elsewhere reinsert the correct gene.
Yeah, and so if you want to go to a different spot to do that, you can take another guide RNA and Cas9, go to the new spot, make a cut, and put the other piece of DNA there.
Gotcha. Thanks.
So how the Cas9 know exactly where he has to cut the DNA?
Where does it have to cut?
No, how Cas9 know where is the location where he has to cut the DNA, like the problem, where's the mutation there is. So it cuts this piece that he doesn't want it. So how it know that?
Cas9 is a non-specific endonuclease in that it will cut DNA anywhere. So the reason this works is because you join Cas9 to the guide RNA. So Cas9 would just chop DNA up randomly, except if it's coupled to the guide RNA, that takes it to the target. The guide RNA is what makes it specific. Cas9 itself is not specific.
So let's take an example for cystic fibrosis, like when we have stop codon. So can we make the Cas9 go cut this piece of nucleotide that's make a stop?
Yeah, so you've got a mutation that makes a stop codon, and you want to get rid of that. So you could make a guide RNA to that region of the genome that has the mutation in it. You could, Cas9 can make a cut there, and then you could take another enzyme to cut out the section you don't want, and then replace it with wild type.
Oh, so who will replace it? So if you want like to put another, we cut this piece of DNA, we want to put another one. Who will replace it? It will not be Cas, because Cas9 is his function is only to cut.
Right, and so what you have now is a gap in the DNA, and you can introduce, for example, a plasmid with your wild type and have a recombination event that replaces that gap where you've cut out the mutation, replace it with the wild type sequence.
Okay. That's clear now. Thank you.
Anything else? Okay, we're almost out of time. The next topic is GMOs, and we don't have time to do that. So we'll finish this section next time. And if there's anything that occurs to you that you want to talk about molecular genetic technologies that we've discussed, we can talk about that next time as well. Or if you have questions right now.
Is this PowerPoint going to be posted to Canvas?
The slide set?
Yes.
Yeah, yes, I'll do that right after the class. Anything else?
Just that thing I was talking to you about before class. The email.
Yeah, I'll check my email as soon as this is over and I'll get back to you.
Thank you so much.
Okay. All right, if nothing else, I'll stop sharing and end the session.
All right, sounds good.
See you all on Wednesday.
See you Wednesday.